Alright, welcome back. In this video, we are going to dive into the actual functions, locations, and structures of the different organelles that we labeled on the animal and plant cell diagrams in the previous video. Before we dive into that, I want to jump back to the previous page. Scroll down to the bottom where we compared plant cells to animal cells, and I want you to add one more thing. I want you to add that animal cells have lysosomes. We're going to talk about lysosomes in a little bit. They are not part of plant cells. I don't think that's something that you're going to be tested on. However, when we do an activity at the end here where we look at the different parts of plant and animal cells and we build a plant and an animal cell, it's going to mention that, and I don't want you to say, we never learned that. So, on top of the next page, the following organelles are present in both plants and animals. Cell membranes. We have a whole topic on cell membranes. It's a very important organelle. A cell membrane acts like a gate. It lets some things out and it lets other things in. It's on the outside of the cell. That's its location. Its structure. The cell membrane is made out of a type of chemical called a phospholipid. And those phospholipids form a double layer called a bilayer. So it's formed by a phospholipid bilayer. Now, that's a lot of vocabulary, I know. The phospholipid bilayer, we have a whole topic dedicated to the cell membrane, but for now, that's what I want you to know. This layer is selectively permeable. You can also call it semi-permeable. We'll define that at the bottom of the page. It controls what goes in and out, and it also helps hold together the structure of the whole cell. Here's a picture of the cell membrane. You see in the cell membrane we have these blue blobs. Those are proteins. Each of these proteins will have a different function. We also have all of these red spheres. These red spheres with the two little tails attached are actually our phospholipids. This is a phospholipid. And you'll see that there is a double layer or a bilayer of these phospholipids. We use this word above called semi-permeable. This is some definitions of level of permeability. Something that is permeable allows anything to move through. A permeable layer is like an open gate. Anything can move through. Semi-permeable, imagine it's kind of like traffic lights. So I'll use green, anything can go through. Semi-permeable, only some things can move through. And impermeable, nothing can move through. The phospholipid bilayer is semi-permeable, which means that it is permeable to some things and impermeable to other things. Here is a diagram of what that looks like. Some things can cross the bilayer. Some things cannot cross the bilayer. That means that overall the bilayer is semi-permeable. This is what the bilayer is like. Some things can cross, some other things can't cross. Here is a little bit of a simulation of a semi-permeable membrane. Oh man, I gotta get my face out of the corner there. Is that this one? There we go. Excellent. So if you are looking at this simulation here, this is, you just got to follow my mouse now. This gray line in the middle is our membrane. If we press play here, we allow particles to move around. We've got two sizes of particles. We've got green particles, which are large, and we've got blue particles that are small. You'll see that all of the blue particles started on the right side here, and now some of them are on the left side. That means that this membrane is semi-permeable. It allows the blue things to cross, but not the green things. And what changes that is how large these pores are. If we increase the size of the pores, then now green particles can pass through. Now it is a permeable membrane because everything can pass through. But if we decrease the pore size so small that nothing can pass through, it is now impermeable. Back to our notes. And I'll put my face back on there. Hi. We're going to go to the next page. The cytoplasm. Both plant and animal cells have a cytoplasm, and prokaryotic cells actually have a cytoplasm and a cell membrane as well. The cytoplasm is all throughout the inside of the cell. It is what makes up the inside of the cell. It is a jelly-like substance. It's not necessarily just like a liquid. It's not just like water. It's mostly water, but it's got some viscosity to it. So if you can think of like honey or jam, it's kind of thick. It's not just flows everywhere. But it does allow the movement. It allows the movement of molecules. In the cell and helps the cell keep its shape. It's this jelly-like substance that fills the whole inside and allows things to move around the cell. Next organelle. This is the one that we had labeled first on both the plant and the animal cell diagrams, the nucleus. The nucleus is in the middle of the cell. Now, it might not directly be in the center, but it's inside the cell. There's only one nucleus for the cells that we're going to look at. There are cells with more than one nucleus, but the cells we're looking at just have one nucleus. It's going to be a spherical shape. Now, here's the important piece about the nucleus. It has the DNA inside, and it controls the cell. DNA is a type of molecule that contains the code for all of the different proteins that make an organism that type of organism. The nucleus has three parts to it. It has the nucleolus, the nuclear membrane, and then the DNA inside. The nuclear membrane is made out of a phospholipid bilayer, just like the cell membrane. And it has pores in it, meaning that it's going to be semi-permeable. Has pores in it. It's going to be semi-permeable, just like the cell membrane. That's going to allow things to move in and out of the nucleus, but it still contains the DNA inside. So the nuclear membrane prevents the nucleolus and the DNA from leaving, but allows some things to move in to access the DNA and then to leave after they've gotten information, or some things to leave. Chromatin is the molecule that DNA is stored as. It's like a form of DNA in a sense. If you take other bio classes, you'll learn more about that structure. But for Science 10, just think about this is the DNA inside here. The DNA has the information to make proteins. The DNA has information to make proteins. And then we have the nucleolus in the middle. This makes RNA, including something called a ribosome, and it's located inside the nucleus. I want you to think that the nucleolus makes RNA and ribosomes. I'm going to color these in red, relating to protein, and you'll see why in a moment. So the nucleolus makes the RNA. The nucleus itself stores the DNA. The DNA is all around this nucleolus here. So where it says chromatin on this little diagram here, you can write in brackets underneath, this is our DNA. And then where we have nucleolus, I will write RNA. I know they're very similar sounding molecules. They are similar in a lot of ways. RNA. Not my best writing. but you get the idea. Next organelle, the endoplasmic reticulum. Well, you can break that word down here. Endo means within. Plasm refers to the shape or the fluid inside the cell. So it's like within the cytoplasm, and it is a reticulum, which is a net. The endoplasmic reticulum has a smooth side and a rough side, or not really a side, a smooth part and a rough part. And it is a bunch of membrane canals that are beginning at the nuclear membrane. In this diagram, that's labeled as the nuclear envelope, but it's the nuclear membrane. And so I'll write here, they begin at the nuclear membrane. And they branch throughout the cytoplasm. Perfect. I was just seeing if my face blocked that label here. If we're looking at this diagram here, the red and closer to the nucleus reticulum is our rough endoplasmic reticulum. Rough, I'm just going to label it as ER. The shortened form for endoplasmic reticulum is going to be ER. That's our rough ER. And then the more orangey color, peach color on the outside, this is our smooth ER. Now, I talked about this when we labeled the cell diagrams in the last lesson, but what makes it rough versus smooth is all of these little bumps on it. All of the little bumps on the rough ER, those are ribosomes. Ribosomes are attached to the rough ER. They are not attached to the smooth ER. This whole system of canals is going to play a couple of different roles in the cell. One of the things it's going to do is it's going to produce protein on the rough ER. So it will produce protein for the cell on the rough ER, and it will produce other substances on the smooth ER. One of the other substances on the smooth ER is lipids. So I'm just going to say, for example, lipids, those are like fat molecules. Those are produced on the smooth ER. Wow, I just am looking at the screen now, and I see that the orange and the red look very similar. It's trying to do orangish for the smooth ER and red for the rough ER and for the proteins. They're also going to move materials around the cell and in and out of the nucleus. So I'm going to say here that they're going to move materials within the cell. It's a system of canals that kind of acts like a highway with little factories attached to it as well that are going to be making lipids, making proteins, and then moving them around inside the cell. It's attached to the nucleus because it will be moving things in and out of the nucleus as well. Ribosomes, we've mentioned them twice now. Ribosomes are made in the nucleolus. They're embedded in the rough endoplasmic reticulum, but they can also be floating around in the cytoplasm. So their location is either on the rough endoplasmic reticulum or in the cytoplasm. They're made of RNA. They're made of a type of molecule called RNA, specifically ribosomal RNA or rRNA, but we don't care about that too much for Science 10. What we do care about is that ribosomes make proteins. Think of them as like little protein factories. Here's a picture zoomed in. This is our nucleus. We have our rough endoplasmic reticulum attached to the nucleus. We have ribosomes attached to the rough endoplasmic reticulum. We have some ribosomes floating around. This is a zoomed in picture of a ribosome. You don't need to know the structure of a ribosome for Science 10. But what you do need to know is that the ribosome makes protein. The ribosome is like a protein factory. And we use proteins for all sorts of things. Proteins are like the functional molecules of your cell. Functional molecules, meaning that they will perform a specific function, like the organelle of the organelles. Ribosomes make these proteins, and these proteins will do different things. Some are structural proteins, meaning that they just form the structure of a specific thing. For example, your hair and your skin and your fingernails are full of structural proteins. There's a protein called keratin that's going to make up your hair as well as your fingernails, as well as, like in other animals, it will make up the horns of like a sheep, for example, which is kind of cool. That's all made of protein. Some proteins you have a defense role. For example, antibodies, which have a role in immunity, are a protein. Your body makes these kind of like a weapon to fight against bacteria or viruses or any sort of invader. Some proteins are important for transporting substances. Hemoglobin, you might have heard of before if you've ever gotten a blood test. Hemoglobin is a protein in your blood that carries oxygen to your cells, and then it can take carbon dioxide as a waste away from your cells. Some proteins are signal proteins or hormones. They're going to control what happens around your body. Insulin is an example of a protein hormone which controls the glucose levels in your blood. Your pancreas will make insulin. It goes around your bloodstream. It tells your cells, hey guys, it's time to grab sugar from the blood and pull it into the cells. Some proteins are used for energy. We can burn proteins if you don't burn your preferred energy sources, which are fat or carbohydrates. Or we have enzymes. Enzymes are a type of molecule called a catalyst, which are catalysts for chemical reactions. You'll learn a lot more about enzymes if you take Biology 20. But this word here, catalyst, it's a chemistry term. It is a molecule, or it could be a substance. It could be several molecules. A molecule that makes reactions happen faster or easier. And it doesn't get used up in the process. The enzyme can keep doing its thing. What I really want you to know for this enzyme piece is that proteins can allow chemical reactions to happen inside your body that would not happen spontaneously or on their own. So they are going to basically work for you to do different reactions. And there's a lot of reactions that they're going to catalyze. Basically, anytime you want to do anything, then like moving a muscle or smelling a smell, there has to be a bunch of different signals that are carried out. It's proteins that are going to be doing the work there. Moving on. The Golgi body. This is one that's present in both the plant and the animal cells. It looks like this, like a stack of pancakes. That's what I'm going to write in this blank here. Looks like a stack of pancakes. It can be easy to mistake the Golgi body for the smooth endoplasmic reticulum. It's not attached to the nucleus, and sometimes there's more than one Golgi body present on a cell diagram. It's going to package materials into vesicles. know if I'm going to spell vesicles right. I think vesicles is like this. Or vacuoles. To be transported. I'm going to write here that to be transported out of the cell. The Golgi body will package up whatever you're trying to get rid of or to transport to a different cell in something called a vesicle and then transport it out of the cell. It is also going to be producing vacuoles and lysosomes. Last note on the Golgi body. On the diagram, I labeled it as the Golgi apparatus. Same thing. Golgi body, Golgi apparatus, same thing. Here's a diagram. These little spheres here are membrane-bound vesicles that have been packaged and that are leaving the Golgi body to go out of the cell. The word Golgi is not a scientific word. It's actually a name of the scientist. It's named after a scientist who discovered the function of this organelle. Mitochondrion. The mitochondrion, the plural version of that is the mitochondria. It's inside the cell. It's an oval that has membranes inside. If you see a diagram and it has squiggly lines inside of an oval, that's the mitochondria. Sometimes these lines are going to loop back on themselves, so it kind of looks like a little mouth there. This is the mitochondria. You can have more than one of them in a diagram on a cell, and you do have more than one inside your cells. Its function is going to be to change sugar as well as other materials that you can respire into a usable energy form. I'm going to say here, usable chemical energy. ATP is usable chemical energy. It is a molecule that we can use to activate different proteins and different functions inside the cell. Cellular respiration is the name of a chemical reaction where we break down glucose. Glucose is a type of sugar to make energy. And the energy form that we make is chemical energy in the form of ATP. Much of the cellular respiration process happens inside the mitochondria. So I'm going to write here, happens inside the mitochondria. If you take Bio 20, you'll learn all the steps to cellular respiration. Some steps don't happen in the mitochondria, but for the purpose of Science 10, cellular respiration happens inside the mitochondria. You're going to break down glucose to make ATP. Final note that I want to write here is that this process requires oxygen. And this is actually the reason that we breathe. This is the reason why we breathe air. We need to take that oxygen into our body so that our cells can make usable energy by breaking down glucose with oxygen in the mitochondria. So if you're out of breath, blame your mitochondria. Lysosomes. Now I wrote at the beginning of this lesson that these are only in animal cells. You'll see that that's not labeled here in your notes. That's only going to be important for the end here. But the words lysis is to destroy or to cut. Some is the body, so it's this sphere full of destruction, really. It is a vacuole filled with enzymes. A vacuole filled with enzymes. Remember that enzymes are our protein catalysts that help make reactions happen. The enzymes that a lysosome is filled with are used to break down wastes or organelles that aren't working anymore. Think of it as your cell's garbage can. Plant cells have something different called a peroxisome, and then some of those kind of waste reduction or waste excretion things are carried out in different parts of the plant cell. But for Science 10, we don't really care about that. I just mention it because it will come up in a bit for an activity. Centrioles. This is the one that I want you to know is only in animal cells. Only in animal cells. You have two cylinders. They kind of look like bundles of pasta, like if you grabbed a fistful of spaghetti to put in the pot, or a bundle of logs. I'm going to say here that it's like a bundle of logs, like if you ever bought like firewood before. The purpose is for cell division. This is for animal cell division. Plant cells don't need these to divide because they actually grow a new cell wall in the middle, but animal cells use these bundles of protein to help pull apart the two new cells during division. Next page, vacuoles and vesicles. These words are very similar and they look similar on a diagram. You will not be able to tell the difference between a vacuole and a vesicle on a diagram unless you can clearly see that one is leaving the cell. Vacuoles store things. And vesicles transport things. But otherwise, they're pretty much the same. They're going to be made at the Golgi apparatus. They look like little bubbles in a cell diagram. And we just wrote here that their function is for storage or transport. If your cell is making something, let's go back to the example of a pancreas cell making insulin. That insulin has to leave the pancreas to go to the rest of the body. You will use a vesicle to store a bunch of insulin, and then you will secrete that insulin out of the cell. Here's the diagram here. Go ahead and try and pick out the cell organelles. This is not my favorite diagram ever. The diagram is unclear about the size of ribosomes. This here is supposed to be our rough endoplasmic reticulum. There's my giveaway here. You can see the smooth side and the rough side, which means that these are all of our ribosomes. So it's going to be really difficult to distinguish between whether or not these little bubbles here are ribosomes or vacuoles/vesicles. Not my favorite diagram, but go ahead and try it out. And also try matching the organelles with their functions. You can pause the video and then you can return to the next page where we'll talk about the special structures for plant cells. Plant cells have some additional organelles not found in animal cells. First one is our cell wall. The cell wall is on the outside of the cell membrane, and it is rigid and strong. It's not flexible like the cell membrane is. And it's made of a substance called cellulose. Cellulose is, here's your fun fact, cellulose is our dietary fiber. If you eat a bunch of plants that have a lot of cellulose, that's where you're going to get fiber from. This is our cell wall. It gives the cell shape and it allows substances such as glucose or proteins to be transported, moved in and out of the cell. So it's not impermeable. It's got holes in it. You can move things in and out. It's not meant to be selectively permeable in the same way that the cell membrane is. Remember, plant cells still have a cell membrane to perform that gate function. Its main thing is it's going to provide that structure for the cell. Plant cells also have a large central vacuole. Those are going to have three main functions. They're going to provide water balance, meaning that they're going to store water. They're also going to provide support and structure. Those are kind of the same thing for the cell, but they help maintain the plant shape through water pressure. This is a plant that has full central vacuoles. If the central vacuoles are full, you see the plant stands straight up and down. This one has a mostly empty central vacuole, and you see that the plant wilts. You say this is a wilted plant. It kind of is falling over. The water pressure for a full vacuole pushes on the edges of the cell wall. So I'm going to say that the full vacuole pushes on the cell wall to provide structure or support. If your plant is low on water, if you have plants at home that wilted, you might know, wow, I better water those. Then it will start to fall over because it doesn't have this pressure pushing on the walls to help keep the plant standing tall. Finally, we have chloroplasts. If you think of plants and you might think of photosynthesis, this is where that happens. Chloroplasts, similar to mitochondria, are going to be oval in shape. But in a diagram, instead of having a squiggly line inside, you're going to see stacks of lines. This is like the most simple chloroplast diagram. But I draw it here because you will probably see a cell diagram on a quiz or a test that you have to label. And not all of the pictures are great, like the picture on the last page. If you have these stacks of lines rather than a squiggle that moves through, that's a chloroplast. Chloroplasts are also only going to be in plant cells. So if you see a chloroplast, also look for the central vacuole and the cell wall just to make sure. The stacks inside are called grana. The singular is granum. That is related to the Latin word for coin. I don't think that's particularly important for you to know for Science 10. But the stacks of them are little discs called thylakoids, which is a funky word. But the thylakoids are these discs. A stack of thylakoids is called a granum. A bunch of granum are called grana. It's a lot, I know. They have a pigment inside the chloroplast that makes them green called chlorophyll. That's a lot of vocabulary around chloroplasts. Chlorophyll is the one that I want you to remember. Don't get chloroplast and chlorophyll mixed up. The way I remember it is that chlorophyll fills the chloroplast. It's the pigment that makes it green. Their function, this is really important. They're going to use water and carbon dioxide to make glucose, to make sugar, through a process called photosynthesis. Oh, that's behind my face. Photosynthesis. Here's the diagram. Carbon dioxide and water enter. The plant uses light energy to create glucose inside the plant, which is not on this diagram. So that's probably important. Inside the leaf here, I'm just going to color this leaf purple, and I'm going to say glucose is made. Besides making glucose, we also release oxygen as a byproduct. This reaction is super important for us because we need both oxygen and glucose to live. Glucose is an energy source, and oxygen is going to be needed to access the energy in that energy source through cellular respiration. The plants don't do it for us. They do it because they also use the glucose through cellular respiration. Remember, they also have mitochondria, but this is an important reaction to know. Here's the reaction on the other page. We're going to balance it because we're good chemists. So 6 carbon dioxides plus 6 water make 1 glucose and 6 oxygen. This chemical here, this is our carbon dioxide. H2O is our water. C6H12O6 is our glucose. And O2 is our oxygen. This is a good chemical reaction to know. Glucose is a type of sugar, which I'm going to write underneath here. Almost done for today. There is a lot of good practice questions that follow on this page, as well as a nice diagram of a plant cell. I'm going to post a link in the description section of this YouTube video to this cell builder activity, which I think is really good for testing your knowledge. If you hover over each of the organelles, it also tells you what its function is. And then you're going to be using it to build a cell. So right now I'm building an animal cell. So I know animal cell needs to have a cell membrane, needs to have a nucleus. It doesn't get a cell wall. I'll give it lysosomes. I'll give it vesicles. And then as I hover over each of these, it will tell me a description of what they do. It's a really good way to test your knowledge and to review the functions of each of these. That's also why I mentioned that the animal cells is the one that's going to have lysosomes, because this activity, that's important for this activity. For Science 10, what's important is that you know that centrioles are only going to be in animal cells. There's also a bunch of good practice questions, which I encourage you to fill out and check your knowledge on. That's all for this lesson. I know it was a long one. Feel free to, it's at the end of the video now, so you made it. But you can go back to specific parts and play it again on faster speed if you need more review. See you for the next one, where we're going to talk about microscopes.